Welcome to Chapter 5. In Chapter 5, we're going to be talking about the plasma membrane. And this is not a boring topic. The plasma membrane is incredibly important. And the reason that it's important is because that's where the cell interacts with the world around it. Okay? So you can contemplate that anything that we have to do in order to interact with our world, the cell has to do the same thing using its plasma membrane. So what do we do to interact with our surrounding world? Well, we eat. So cell nutrition is based on the plasma membrane being able to take the nutrients necessary for the cell to live. We eliminate waste. So the plasma membrane is where the cell chooses to excrete whatever waste it is no longer using. Um, we maintain homeostasis. And as we've said before, everything within our bodies have to be within narrow functioning limits. This is also true of cells. Cells need to be maintained within narrow homeostasis, within narrow functioning limits. And as a consequence, if there's too much inside of something in the cell, it needs to go out. That goes through the plasma membrane. If there's an essential nutrient or something that needs to come in, that goes through the plasma membrane. Okay. What else do we do? We taste the environment. We smell it, right? Cells smell and taste the environment using receptors on its plasma membrane. We talk to each other. Cells communicate to each other via glycolipids on a cell membrane. What else do we do? We maintain integrity. Imagine this. On top of the plasma membrane, functioning as a mouth, and as a site of excretion, and as a site of smelling and tasting, it's also the part that keeps the cell together. Wow, that's incredible. Because you're asking for two things to happen. You're asking for an object to be sufficiently dynamic, sufficiently pliable, sufficiently fluid, so that this exchange can happen between the cell and its environment. But at the same time, this plasma membrane has to be strong enough that it won't allow your cell to rupture. Imagine your body is made out of 3 trillion cells, each one of them wrapped by a plasma membrane. And we walk around and they don't fall apart. So imagine, I mean, when you start to understand it, you start to realize how incredible this is. So the plasma membrane has to be fluid enough to allow things to come in and go out, but strong enough so that when you and I are jogging or running or run into something, cells don't just burst. Because at the end of the day, the plasma membrane is something that's incredibly dynamic, incredibly fluid. And to conceptualize that it can be that dynamic and that fluid, yet that uh, able to restrain and to accept shock and accept trauma is just incredible. So the plasma membrane is just an incredible structure, essential to the basic building block of life, the cell. Okay. So what is it? What is it built out of? Four main items make up the plasma membrane. The main component is what we call the phospholipid bilayer. This should be reviewed to you. Okay. Hopefully you remember that the phospholipid, you have a sugar backbone, glycerol. You have 2 fatty acids, and then on the other side, you have a phosphate group, and then you have a random sugar, a, 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 a sugar of some sort, whose sole purpose is to serve in cell-to-cell recognition. Okay, we've covered all this before. So that's what we are mainly made of. The majority of the plasma membrane is made of this so-called phospholipid, and it's a bilayer because it's not one layer, it's two. As a matter of fact, if you're to able to separate one from the other, and some researchers do, that's called a leaf. Okay, so a leaf is one half of a phospholipid bilayer. The second thing that you're going to find is cholesterol, which is very, very important. And we're going to address why that is in a second. The third thing that you're going to find, which is very, very important, is proteins that are embedded in or lie on the bilayer. So there's two different types of proteins. Okay, and we're going to address those regarding on how they lie in the plasma membrane. They either lie inside the plasma membrane or they adhere just outside the plasma membrane. So geographically, there are two main types of plasma proteins. And then the fourth one is the short carbohydrate chains or cells on the surface. Our author likes to call it the glycocalyx. I don't like that. I don't. Very few people use that term. We use it in microbiology because certain bacteria have a well-defined glycocalyx. There's some protozoa that have a glycocalyx. There's definitely algae that have a glycocalyx. We don't really have a glycocalyx by definition, okay? What we have is we have glycolipids. Let me try that one more time. Glycoproteins, proteins with sugars on them. And the whole purpose of this thing is cell-to-cell recognition, okay? Notice here that it says that the function of the glycocalyx is cell adhesion and binding sites. Right there, that should let you know that that's not us. A glycocalyx is what allows bacteria to adhere to a surface very well, okay? But for you and me, when cells come together to form tissues, they don't use a glycocalyx to adhere to each other. They use gap junctions. They use proteins that tightly tether one cell to the next. Okay, but we're going to flow with what the author says on this one, and we're going to say that this sugary outer layer is called a glycocalyx. Okay. So, let me move me out of the way. Okay, so here's our phospholipid bilayer, and hopefully you remember that again it consists of a glycerol backbone right here. Actually, I don't need to do this. Look, is right. What is this? I don't know what this is. This is very perplexing. Let's try it again. So here's our phospholipid molecule, okay? And again, this is review. We've addressed all those before. Here's the glycerol backbone. Here's the phosphate, okay? That phosphate is very electronegative. Hopefully you remember that. And electronegative means that this guy has a net negative charge. See that? Okay. And then there's two nonpolar tails. And then here's our variable sugar group at the top. So consequently, we have what we call a hydrophilic or water-loving head and a hydrophobic or water-fearing tail, okay? And that's your phospholipid. So when we look at this, each one of these is a phospholipid molecule, and notice that each one of them has two hydrophobic tails and a hydrophilic head, which is a phosphate group with that variable sugar. Sugars have a net negative charge also. If you've forgotten, sugars have hydroxides, and hydroxides have a net negative charge. So if you're to divide this like this up, that would be what's called a leaf. So this would be half the leaf, and this would be the other half of the leaf. And one of the things that your author goes to great extent to explain to you is that this arrangement that we're seeing here, this bilayer, is not a what's the word I'm looking for? Is not a coincidence. It is not a random event. It's something that happens because we have the inside. Let's see, which way are they saying is the inside? So this is the inside of the cell, and this is the outside of the cell, right? And there's one thing that is common to both sides of the cell, and forgive me, of the plasma membrane. It was common is that it is a water environment. So the inside of the cell is a water environment, and the outside of the cell is a water environment, okay? In science, we call that an aqueous environment. So this is an aqueous environment here, and this is an aqueous environment here, okay? Why do we care? We care because the arrangement of this phospholipid bilayer, it arranges like this because the polar heads want to associate with the aqueous environment outside of the cell, but the polar heads on the other leaf want to arrange with the aqueous environment or hydrophilic environment inside the cell. As a result, we end up with this phospholipid bilayer because we have water both outside and inside. Water is polar, if you remember that, water is polar. So therefore, the hydrophilic heads are always going to associate with the water. Okay, very, very neat stuff. Okay, so let's go back. So here's a phospholipid bilayer. And what's the second thing that we have? Let's see. Let's make sure we're not missing anything. A double layer of phospholipid molecules whose hydrophilic heads face outward. They face outward. And whose hydrophobic tails point inward towards each other. Okay? So to recap, this is hydrophilic, but the tails are hydrophobic. Okay? What's the second thing that we have? The second thing that we have is cholesterol. So you can see that we have cholesterol here, 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 here. Okay? So cholesterol molecules that act as a patching substance and that help the cell maintain an optimal level of fluidity. Okay? Actually, the patching part, secondary role. The primary role is maintaining cell fluidity. Okay? So I don't know if you guys remember when we were talking about this last time, but when you have a hydrocarbon, such as these nonpolar tails, they can either be saturated or non-saturated, okay? How can we tell the difference? If they're saturated by hydrogen, they're straight like this guy right here. If they're not saturated and they're missing some hydrogens, they have a double, sometimes a triple bond, and you end up with a kink in the middle of the tail like that, okay? Well, if you and I insist on consuming nothing but animal fat, and that animal fat is saturated fat, and it typically is, how do we know? Because saturated fat is solid at room temperature, okay? Now, why is it solid at room temperature? Because when it's saturated, you have nothing but straight tails like this one right here. And we have nothing but straight tails. One straight tail will associate with the second straight tail, okay? In chemistry, we call these van der Waals interactions, okay? They're very weak. As a fact, they're the weakest interaction between atoms. However, if you have a long tail, and we do, well, sometimes we have about 30 to 40 carbons, what starts out as a weak interaction ends up being a powerful interaction. So if you have a whole bunch of phospholipids that are all saturated, what happens? The tails start sticking together. Do we want that? No. By, by definition, the plasma membrane is supposed to be fluid. It's supposed to be dynamic. So if your phospholipids are sticking together and they're turning into butter, butter is not very dynamic. It's not very fluid. So consequently, your cell is going to have difficulty doing what it likes to do, which is take nutrients in and get rid of wastes, things that it needs a dynamic fluid membrane for. Okay. So how does our body fix this problem? Well, one way that it fixes it is that your liver produces cholesterol. Okay. And if you remember from last time, cholesterol is a hydrophobic molecule. It has 4 rings to it. Okay. And because it's hydrophobic, it can associate with the hydrophobic interior of the plasma membrane. But what this cholesterol does when it comes to the membrane is it introduces randomness. So now where you usually have these phospholipid tails like this, the cholesterol gets in the middle and it pushes them apart. It introduces randomness, okay? And in introducing that randomness, it forces the plasma membrane to become more dynamic, more fluid. So that's the main role of cholesterol, is to maintain an optimal level of fluidity in the plasma membrane so that the cell can do what it needs to do, okay? So you might think to yourself, I don't get it. Then why is cholesterol so bad? Well, cholesterol is bad because it will adhere hydrophobically not only to plasma membranes but also to itself. So cholesterol can pack onto itself in your arteries and make plaques in there, okay? And that can be very dangerous. So you need to lower. We're going to nutrition now. So you need to lower your cholesterol, okay? But if your body needs cholesterol, this is what's interesting, okay? Everybody's like, well, just switch your diet. Yes and no, okay? If you stop consuming cholesterol and your body needs cholesterol because your membranes are very non-dynamic, you know what's going to happen? Your liver's going to make cholesterol. So you're like, what? So I'm on non-cholesterol medication and my liver's going to create cholesterol? Yep. Because your body needs it. There's no way around it. So how do you lower the incidence of cholesterol? Well, amusingly, the answer is diet. Okay? What you do is that when you consume fats, you make sure that you consume fats from plants or from fish. When you consume omega-3 fatty acids, okay, what that's telling you is that you're consuming a fatty acid that has many double bonds in it. And so if your body can make phospholipids that are themselves random and increases the fluidity of the plasma membrane, it will not need this cholesterol, and consequently your liver will stop producing it. Does that make sense? So there should be no wonder that the longest living people in the world live in Japan. What is their diet consist of? Usually it's a little bit of rice or carbohydrates, but mostly it's steamed vegetables and some type of fish. I mean, their diet is fantastic. And because of this, they don't need so much cholesterol, okay? But the main role of cholesterol is to optimize the fluidity of the plasma membrane so that this membrane can do what it needs to do. not be rigid, because if it's rigid, it cannot, again, assimilate what it needs and excrete or uh, get rid of whatever it needs to get rid of, okay? Eliminate is the word I was looking for. What else does it have? It has proteins. Proteins which are integral, meaning bound to the hydrophobic interior of the membrane, or peripheral, meaning not bound in this way. So, did you guys catch that? This is important. You can either have an integral membrane or a peripheral membrane. So let's take a look at this. Here is a structural protein. How do I know? Because here's a cytoskeleton. So essentially, the cytoskeleton is making a scaffolding and has put a rivet, effectively, on the plasma membrane at this site. Does that make sense? So we have, and you know which one it is. This is the intermediate filaments, right? The least dynamic of them all. We covered this, what, last week? Okay. So here's the cytoskeleton. And we have a protein that is inside the plasma membrane. Do you see that? If it's inside, that's called an integral protein. Why does that matter? It matters because this protein has to be hydrophobic in order to embed itself inside the plasma membrane. Remember, as we keep on saying, by the time I'm done with you, you're going to be tired of me saying it. But likes dissolve likes, okay? So if we have a hydrophobic environment here, which is all these tails right here, that means that this protein, if it's going to be embedded in here, it has to be hydrophobic. And it gets very elegant, okay? And we're going to hit that in just a second. But in order for this protein to be happy with these lipids, all the amino acids that make the exterior of this protein, of this integral protein, are hydrophobic, okay? Now look at this protein over here, just to the outside. Do you see the second protein? That's a peripheral protein. How do we know? Because it's not embedded to the inside of the plasma membrane. So what do you think the character of the amino acids are here? Do you think they're hydrophilic, seeing as this is an aqueous environment? Isn't that cool? So this rivet, to hold the cytoskeleton in place, consists of not one, but two proteins. One integral protein that is hydrophobic, and one, what integral protein, I said that, right? Sometimes I wonder about myself. One integral protein, which is hydrophobic, and one peripheral protein that is hydrophilic. And they're both docked in such a way that the cytoskeleton can grab a good hold of it. Okay? So that's one role of proteins, structural. Okay? Actually, I'm getting ahead of myself. This is a very interesting topic for me, so I love getting ahead of it. Let's see. Oh, okay. See, I'm glad that I stopped myself. So this is important, okay? So here's our phospholipid bilayer. We'll talk about proteins in a second again. Here's our phospholipid bilayer. And here we have, it says hydrophilic molecules pass through freely small nonpolar molecules, okay? So, so these two mean the same thing. Hydrophobic molecules, okay, and small nonpolar molecules. Those two mean the same thing. This is two different ways of saying the same thing. Effectively, this is small and it's not charged. So if you have a small nonpolar molecule or a hydrophobic molecule, it will traverse the plasma membrane without difficulty, okay? So you might go, well, isn't the hydrophilic head's going to stop it? Not really. This is a little head, right? It's just a little guy. Well, this is like 30 to 40 carbons in length. So this is a lot of real estate in here, and these heads are not really going to do much. So if you're small and you're nonpolar, you're going to have no trouble going through that plasma membrane. By contrast, if you're hydrophilic, in other words, a charged molecule, sugar, ion, whatever, okay, you're not going to make it through. You're just not going to make it through. You're going to make it through the polar heads, and then as soon as you get a little bit in, the hydrophobic environment is going to push you back out. Okay, so hydrophilic molecules do not pass through freely. What else? Also, even large hydrophobic molecules make it through here. How do we know? Cholesterol is pretty big. The problem is that if it's big, it might get stuck in the plasma membrane. So that's one problem. Now, why am I bothering you with this? Well, this is very important for a couple of reasons. Number one, your body uses a lot of molecules to communicate with each other, with itself. Okay, these molecules are called hormones, and hormones can sometimes be lipid-based. We talked about this. For example, testosterone, right? Estrogen, those are lipid-based. Okay. So I ask you, if you have a lipid-based hormone, what's it gonna do? Is it gonna be impeded by the plasma membrane? Well, the short answer is no. No, it's not. It's going to be able to traverse right through the plasma membrane. So where does the effect of these hormones take place? Incredibly, the answer is in the nucleus. Why? Because it has absolutely no problem going through the plasma membrane. That's pretty incredible. By contrast, if you have a hormone, and that hormone is made out of amino acids, and those amino acids are particularly polar, that hormone is going to try to go in, and is going to access denied. It's not going to make it in. Okay. Okay. Well, what do we have to do then? We have to do this thing called signal transduction. Okay, which means that you have a beautiful protein here, and that protein is going to receive this hydrophilic hormone. The protein is going to change shape, and then it's going to transduce or transport that that message from the outside of the cell to the inside of the cell. But the hormone doesn't make it inside. It can't, because it's charged. Is that incredible? Okay, so that's hormones. How about medication? If you're taking a medication, not all medications are made the same. Some medications are hydrophobic. If that medication is hydrophobic, it's going to be able to enter your cells no problem, because it's going to be able to go through the plasma, the phospholipid bilayer. But if the medication is hydrophilic, water-loving, it's not going to go into the cell. It's going to have the same mechanism as a hormone. It's going to attach itself to a receptor, a protein, and then that protein is going to carry out signal transduction. In other words, it's going to grab the message and it's going to transfer the message to the inside of the cell, but that medication is never going to make it inside the cell. So how things act on the cell and where those things act in the cell depends on the polar character of those things. If it's hydrophobic, then that messenger is going to act inside the cell. If it's hydrophilic, then that messenger is going to act outside the cell through signal transduction by a protein. Very, very cool. Oh, I almost forgot. And then there's water. Water has its own rules. It just does what it wants to do. By definition, water is a small polar molecule. But as it turns out, water can actually traverse the phospholipid bilayer. It doesn't do it very quickly, but it does do it. And it's counterintuitive. you ask a scientist, they'll walk away. We don't really know why. But we have a polar molecule, which is water, which apparently goes, I'm just going to violate the laws of physics and go through this through this hydrophobic environment. But all we can tell you is that it happens because we know that it happens. Okay. Let's see. Oh, this is very interesting. Oh, this is so interesting. I love this because I didn't learn this until I was in graduate school, and then it blew my mind. Okay, so one of the things that your author is trying to convey to you is how incredibly dynamic, mobile, fluid this plasma membrane is. Okay, because you look at the picture and you're like, yeah, it's a bunch of things just standing there. Oh no, these things, they're rotating, they're spinning in place, they're flipping back and forth, they're diffusing across the plasma membrane. The movement of these phospholipid molecules with each other is just mind blowing. Okay, so let's see, because of their character, phospholipids traverse the plasma membrane frequently. So every 2 to 3 seconds, if you could put a little like green fluorescent protein here and make this guy fluoresce so you can see it under a microscope, you would see that every couple of seconds or 3 seconds, this guy would go boop and go to the other side, and then go boop and then come on this side, and then go boop and come over here. So very, very, very frequently, these things just go back and forth because really that polar head is not sufficiently big enough to prevent it to go through the hydrophobic environment on the inside. So these things are just popping back and forth all the time, and they're spinning in place, and they're vibrating and moving all over the place. Okay. As a matter of fact, they did an experiment, and they put green fluorescent protein on one of these things, and they put a laser on it. Okay, and then they measured how long it took for this thing to diffuse throughout the entire cell. 3 seconds. Within 3 seconds, a phospholipid will diffuse and cover the entire surface of the plasma membrane. If that doesn't blow your mind, then I don't know what else to tell you. So, anyways, because of their characters, phospholipids traverse the plasma membrane very frequently, and they can diffuse laterally, covering the entire membrane in literally seconds. Very, very neat. Okay. Then we're talking about cholesterol here. Let's see. There we go. In animal cells, the cholesterol molecules are interspersed between phospholipids, and the plasma membrane perform two functions. We've already covered them. They act as patching material that keeps some small molecules from moving through the membrane. Uh, I really hate that statement, but the reason they call it patching material is because, again, the cholesterol is hydrophobic, so it has a tendency to pull the tails towards itself. So yes, it functions like this. But really, like you and I said, the key role is to keep the membrane at an optimal level of fluidity. Okay. All right. Now we're going to go repeat where I had already left off, which is integral proteins are responsible for nutrient transport because they span the phospholipid bilayer. Okay. So apparently, yeah, we will get there. I'm becoming frustrated, but we're going to get there. Okay. So again, if you need something to go into the cell or out of the cell, you need something that spans the cell membrane, right? So we're looking at something like this guy right here, although that's a receptor, okay? But you need something like this that's a channel, okay? And just when you think things can get any more complicated, channels, these proteins have to have a hydrophobic exterior so they can associate with the plasma membrane, and then they typically have to have a hydrophilic interior so that the substance that is traversing the channel can go through the middle. Very, very cool. Okay. So, but the point is, if the cell needs nutrition and it's taking in, let's say, yummy glucose, okay, that yummy glucose requires an integral protein because that integral protein spans the phospholipid bilayer, right? If the channel looked like this, it wouldn't work, right? The glucose would come in, boop, and then it gets stuck there. It has to span, has to cross the phospholipid bilayer, okay? So you need an integral protein, and that's what that slide is telling you, okay? Okay, so integral proteins are responsible for nutrient transport because they span the phospholipid bilayer. This slide is in blue because I find it amusing. You know, I have to look at my slides before I talk to you guys. And I looked at it and it threw me off. In animal cells, proteins are interspersed between phospholipid molecules in the plasma membrane, and they perform two functions. But your author says cholesterol molecules. This is wrong. So, and this has been wrong for so many years. Their quality control is not so good, I tell you. But this is what the PowerPoint said, and I leave it in blue because I find it entertaining that they make huge mistakes and nobody catches them. Anyways, the point is, in animal cells, proteins are interspersed between the phospholipid molecules in the plasma membrane, and they perform 4 functions, not 2. So there's 2 things wrong with this slide. 4 functions. They perform structural support, which we just covered. They perform cell identification by serving as external recognition protein that interacts with the immune system cells. They carry out communication by serving as external receptors, and transport by providing channels. So how's that go? A picture's worth 1,000 words. Here they are. Here's a 4. This is what you need to know, okay? They can serve as structural proteins. Remember, here's our cytoskeleton. So structural means structure. It maintains the cellular structure. It either holds the cell in a certain shape or it holds organelles within the cell in a particular place, okay? That's a structural support. So membrane proteins can provide structural support often when attached to parts of the cell scaffolding or cytoskeleton. And again, remember, if it's part of the cell cytoskeleton, that's intermediate filaments. Recognition, okay? So here, this is supposed to be, well, this is supposed to be a cute little sugar right here. And let's see, protein fragments held within recognition proteins can serve to identify the cells as normal or infected to the immune system. So I stand corrected. The author is not addressing cell-to-cell recognition. It's addressing cell-to-immune system recognition. So this is an advanced type of recognition, which is called antigen presentation, okay? So what happens, and I'm not going to spend a lot of time on this because we're supposed to be touching, no pun intended, but the surface of the topic, is that, you know, if you have a bad guy in your body, a cell or a virus, okay, you have cute little soldiers by the name of monocytes that are in vigilance 24/7. And anytime that one of these monocytes runs into a bacteria or a virus or anything that just doesn't belong there, it consumes it. It's called phagocytosis, cell eating. And then it adds digestive enzymes to it. We've covered this. That's in the organelle called a lysosome. And then that material is broken into small pieces to render it inert. But some of the pieces are presented with a protein and that's called antigen presentation. Very cool, okay? Because effectively, your monocyte, which is a soldier, just caught a bad guy, it killed it, and then it kept part of the uniform and went, hey guys, if anybody runs into one of these guys, they don't belong here, feel free to kill them. Scary, right? These guys don't get around, okay? But that's how you and I stay healthy, is that we have these very vigorous monocytes keeping us healthy all the time by doing protein presentation. It's actually called antigen presentation, okay? So that's an important role of proteins. Another one is communication. So earlier when I was talking about signal transduction, this is what I'm talking about. See this little H right here? That's a hormone. Is that hormone hydrophobic or hydrophilic? If you guessed hydrophilic, you're correct. Because remember, if this hormone needs a protein to converge to the inside of the cell, then that means it cannot go through the plasma membrane. Because the only thing that goes through the plasma membrane is a hydrophobic hormone, right? If you had a hydrophobic hormone, it go right through. This one, therefore, must be hydrophilic, and the only thing they can do is associate with a receptor. This whole thing is a receptor, okay? And this receptor is carrying out, remember my $20 word, signal transduction. In other words, it's transducing a signal, forgive me, from the outside of the cell to the inside of the cell. See these little waves in here? It's telling the inside of the cell that something's happening over here. So receptor proteins protruding out from the plasma membrane can be the point of contact for signals sent to the cell via traveling molecules such as hormones that are what? Hydrophilic. And then finally, a channel. This is a simple channel. Not surprisingly, your author chose yummy low glucose. That's usually what cells are moving around. You gotta have energy, you gotta have yummy low sugar, right? So here's the outside, here's the inside of the cell, and this is a transport protein. We transport a lot of things. Sugar just happens to be one of them, okay? You transport a lot of things through the cell membrane. And one of the things that's interesting about this, again, is that the outside of this plasma membrane has to be hydrophobic, so it can attach to the hydrophobic interior, but the inside has to be hydrophilic, because remember, sugar is polar and it only associates with hydrophilic environments. So very, very interesting, okay? So proteins can serve as channels through which materials can pass in and out, okay? And we're gonna go more into detail into this. This gets really interesting. But these are the main four: structural, recognition, communication, transport. Okay, so it says the plasma membrane today is described as a conceptualization called the fluid mosaic model. It views a membrane as a fluid phospholipid bilayer that has a mosaic of proteins either fixed within it or capable of moving laterally across it. I don't think this does it justice. Do you? Let's go over here. I don't have enough pages open. Let's open one more. Let's type in mosaic. There we go. This is what I want you to picture. You picture a plasma membrane. That's what a mosaic is. Nobody knows what a mosaic is here because really it's kind of a Middle Eastern thing. Okay. Some of you have stuff like this in your bathroom. Okay. But this is a mosaic. So imagine, okay, that this beautiful mosaic is the surface of your plasma membrane. You know what you're seeing here? Not the phospholipid bilayer. What you're seeing is proteins. There are so many proteins on the surface of the phospholipid bilayer that you almost can't see the phospholipid bilayer. You literally have a mosaic of different proteins, and it would look something like this, something like that, a randomness like that. Why? Because you have a whole bunch of different structural proteins, and you have a whole bunch of different recognition proteins, like a lot. Okay, many. Every time you kill a bug is a different recognition protein. How many bugs does a monocyte kill a day? Your guess is as good as mine. I'm willing to bet it's more than one. Okay, plus it has to tell everybody that it's part of our body, so that's another recognition protein. And then communication. Oh my goodness, there's got to be hundreds of hormones telling each cell what to do every minute of every day. So you have 100 different receptors and the different things that need to come into and out of the cell. There's got to be 100 or more transport proteins. Okay, so by the time you're done putting all the proteins that you need on the surface of the cell to carry out the function of the cell, you end up with something looking crazy like this. You're like, huh, I don't even see the phospholipid bilayer. All I see is proteins. Yep, that's about right. That's all you're seeing is a whole bunch of proteins. Okay. Now imagine that it's in a plasma membrane that's doing this. It's moving around because it's dynamic, highly dynamic. If you're getting seasick, then I'm getting the point across. I don't know why I moved myself so far down. Let's go back. Okay, very, very cool. So, the fluid mosaic model. It views a membrane as a fluid, okay, phospholipid bilayer that has a mosaic of proteins either fixed within it because they're structural or capable of moving laterally across it because they're doing everything else. Okay. Next one, diffusion, gradients, and osmosis. If I were to name this, I would go gradients, diffusion, and osmosis, but oh well. So diffusion, very important. The movement of molecules or ions from a region of high concentration to a region of low concentration. What's a good example of this? Oh, you know what? I need to take a little break, and I know that I can, so I'm going to. I'll be right back in a second. Okay, I'm back. So anyways, diffusion. So examples, oxygen in the lungs, okay? This is incredibly interesting to me because the whole event is passive. What does passive mean? Passive means that it does not require energy, okay? So for example, why do my cells receive oxygen from the capillaries? And the answer is because the cells have less oxygen in them than my capillaries. Okay. Why do my capillaries receive oxygen from the arterioles? Answer, my arterioles have more oxygen in them than my capillaries. Huh. Why do my arterioles receive oxygen from my arteries? Because the arteries have more oxygen than the arterioles. Why does the oxygen go from my lungs to the arteries? Because I have more oxygen in my lungs than I do in my arteries. Why does oxygen go into my lungs? Because there's more oxygen in the atmosphere than there is in my lungs. What? Think about that for a second. That means that oxygen goes from the atmosphere to my lungs, to my arteries, to my arterioles, to my capillaries, to my cells, and no energy is being used. Okay? All that's happening is I'm inflating the lungs. That's it. That's it. Oxygen, all it's doing is it's diffusing from an area of high concentration to an area of low concentration. That's all it's doing. Incredible. Okay? Um, Same thing can be said about the carbon dioxide. The carbon dioxide is coming out because it is diffusing from an area of high concentration to an area of low concentration. Okay. Now it gets even more interesting if you contemplate that we never have to play with the system. So if you are walking in a relaxed manner, the gradient between your lung and your arteries is going to be shallow. Therefore, the rate of diffusion is going to be slow. But now you're walking at night and I don't know, the clown from It jumps in front of you and you find yourself sprinting home, right? What happens? Now, your oxygen levels in your cells are down here and the atmospheric oxygen is still where it started. The gradient has increased, right? It has increased because your cells are using more oxygen. What do you think happens when that gradient increases? The answer is that the rate of diffusion increases. So because the demand in your cells has increased, the rate of diffusion into the cells through that entire system has also increased. Nobody has to tell Mother Nature to hurry up. If you need it, it's there. If you don't need it, it's not there. The laws of physics are beautiful. So, same thing in a little tree, right? You have a little tree, you have diffusion of minerals in the xylem going up in the tree. Okay. If the tree is moving a lot of water up its xylem, and this is a passive event, that means that it's not using energy. If it's moving a lot of water, it sucks up more minerals. Okay. The more water, the more minerals, the healthier the little tree. Okay. But the point is that you don't have to speed it up, you don't have to slow it down. Mother Nature, the laws of physics are set up in such a way that diffusion immediately solves need based on that gradient. Okay. So a gradient, therefore, okay, if diffusion is moving of something from a high concentration to a low concentration, then a gradient is the difference in concentration. The greater the gradient, the greater the rate of diffusion. The less the gradient, the less the rate of diffusion. Very, very cool. Okay. Okay, a concentration gradient defines the difference between the highest and the lowest concentrations of a solute within a given medium. Essentially what I just said. Through diffusion, compounds naturally move from higher to lower concentrations, meaning down their concentration gradient. So that brings me to my little kiwi drink here. Okay. And I don't know if you can tell here. Let me move this thing out of the way. I don't know if you can tell, but I have green on the bottom. Then I have yellow here. And then I have purple here. And then I have blueberries up here. That looks tasty, doesn't it? Why am I showing you this beautiful thing? Because if you go to a nice little, forgive the pun, watering hole, you might ask for something that looks like this. How do they do this? Well, the concentration of the green liquid is greater than the concentration of the yellow liquid. And the concentration of the yellow liquid is greater than the concentration of the purple liquid. And the concentration of the purple liquid is greater than the concentration of the blue liquid, right? And then they pour this nice and slow, and then as it sits there in front of you, what takes place? Diffusion. So the green runs into the green runs into the yellow turns into the purple runs into the blue, and you end up with this nice little stratification of colors. Okay. So through diffusion, compounds naturally move from higher to lower concentrations, meaning down their concentration gradients. There. Now that I've completely blown everybody's attention by thinking about a delicious little drink, let's move on. So here's diffusion. This is a way that we like to teach it. Okay. If you decide to do this at home, guys, let the girls do it. We don't know what we're doing. If you give a dye dropper food coloring to a young lady, she'll put 1 or 2 drops in there. If you give it to a guy, he'll put like 3 squeezes. That's not going to work. You can't do that, guys. You can't do that. You put a couple drops, okay? If you put a couple drops, then with time, you can see diffusion take place until you end up with a homogeneous solution, okay? And I'm going to tell you, your author calls this equilibrium. He's almost right. This is called dynamic equilibrium. And the reason for this is because the water molecules do not stop moving at any time. So let's say, for the sake of argument, you had a membrane here. You would have molecules going one direction, and you'd have molecules going the opposite direction. But the concentration would be identical on both sides. Because the concentration is the same on both sides, thus equilibrium. However, because molecules are actually moving from one point to another, it is dynamic. And consequently, we have what's called dynamic equilibrium. Okay, now we're moving to a couple other things. So we're going to hit all these in a little bit more detail in the next section. But energy must be expended to move compounds against their concentration gradient, meaning from lower to a higher concentration. So diffusion, we've been saying, is the movement of a solute, or moving of something from an area of high concentration to an area of low concentration. Can you move things from an area of low concentration to high concentration? The answer is yes, but because you're swimming against the current, it's going to take energy. Okay? It's going to take energy. We need to define what a semipermeable membrane is. A semipermeable membrane is one that allows some compounds to pass freely while blocking the passage of others. So we're going to cover why that's significant in a second. Now, water is so important that the diffusion of water has its own name. So osmosis is a net movement of water across a semipermeable membrane. So semipermeable membrane would imply that it allows water to traverse the membrane and nothing else, from an area of lower solute concentration to an area of higher solute concentration. Okay. What? Well, you just gotta, this is not that complicated. Just keep in mind what is moving, okay? Remember, osmosis is water. So the diffusion of water is the movement of water from an area of high concentration to an area of low concentration. If you remember from last chapter, a solution consists of the solvent, water, and the solute, what's dissolved in the water, okay? So if the water concentration is high, that means that the solute concentration is low. If the water concentration is low, that means that the solute concentration is high, okay? So if I was to say that osmosis is a net movement of water across a semipermeable membrane from an area of high concentration of water to low concentration of water, I would be correct. But if I was to say what this slide says, across a semipermeable membrane from an area of low solute concentration, i.e. high water, to an area of high solute, i.e. low water concentration, it would also be correct. And in case you're not confused yet, you can also say it a third way: osmosis is a net movement of water across a semipermeable membrane from a hypotonic environment to a hypertonic environment. Okay, so we're going to hit that in a second. Okay, hypo means below. So hypotonic means low solute, low solute. So the movement of water from a low solute or hypotonic environment to a hyper means high solute environment. Hopefully you guys are doing okay. Effectively, this is vocabulary salad, okay? 3 different ways of saying the same thing. All you have to keep in mind is that osmosis is looking at the water concentration. Osmosis is literally the diffusion of water from high concentration to low concentration. And if that is the case, then these 2 statements are also correct. Okay? Ooh, didn't go down. Let's try it again. Oh. So here we're going to demonstrate what happens when you put a solute. So what we have here is we have 2 beakers, and then in the middle we put a semi-permeable membrane. Okay, and that means that this membrane is permeable to water, but not salt. Okay, and now what we're going to do is we're going to put salt in one of the beakers and not the other. Okay, well, what's going to happen, okay, is that through osmosis, water is going to move from an area of high concentration to an area of low concentration. Why is this low concentration? Because over here we have nothing but water, and over here we have water plus salt. So the water wants to move from high concentration to low concentration until what? Well, typically the answer is until both concentrations are the same. However, because there's such a thing as gravity, okay, we're going to have osmotic pressure going in this direction, but we're going to have gravity pushing back. So there's going to be a point of equilibrium where the force due to gravity is equal but opposite to the force due to the osmotic pressure. And at that point, this is going to stop moving. And guys, if you're interested in seeing this, no, you're not. It's a little bit like watching grass grow. It takes forever, okay? The only thing I find amusing about this, when I've done in the past, and it doesn't work so well either, is to use different salt concentrations here and see that at higher salt concentrations, the water column goes higher and higher. And even that doesn't work very well sometimes because, you know, the laws of physics, you know, they don't work perfectly all the time. But that's what's supposed to happen, okay? Osmotic pressure goes in one direction through this semi-permeable membrane, and it's going to push the water column up until, well, until gravity pushes back. Okay, we need to go, here we go, this is where we need to go. Okay, so what we're going to do is we're going to put a cell, here's a cell, here's an animal cell, here's a plant cell, okay? And what we're going to do is we're going to put these cells in different solutions, okay? So imagine a beaker here, imagine a container, and in this container we have put a hypertonic solution. Hyper means above. In other words, the solute concentration in the water in this beaker is above this, the solute concentration in the cell. Okay, that's hypertonic. This cell is in a different beaker, different. And in this beaker, the solute concentration in the surrounding water is equal to the solute concentration inside the cell. When that is true, this is called an isotonic solution. Okay, iso means the same. So the same outside and the same inside. In this one, this is a third beaker, okay? And here, this is going to be hypotonic. Hypo means below, so the solute concentration is going to be lower outside than inside, okay? And that's the same thing over here with these plant cells. So what happens in the hypertonic solution? Well, there's more solute outside than inside. So what is osmosis going to want to do? Remember, water, osmotic pressure, water wants to go from high water concentration to low water concentration. So if there's more solute outside, the outside is hyper, then there's more water inside than it is outside. So the water is going to want to come out of the cell to the outside, and the cell is going to shrivel up. Okay? If you had a blood cell, it would shrivel up so much that at the end it would do the same thing that this guy does. It'll break up. It'll implode. With a plant cell, it kind of goes down, and the shape of the cell is somewhat held together by the fact that there's a cell wall there, but there's no water, so there's no trigger pressure, and the darn thing falls over, right? Here we have an isotonic solution. So again, the concentration of solute is the same outside as it is inside. So what do we have here? Remember, dynamic equilibrium. So we have water going in and we have water going out. But when you have water going out, the same amount comes in. That's an isotonic solution. So your animal cell is going to be happy and your plant cell is going to be happy. Okay? Notice the turgor, see the plant, it's all happy. In the third one, we have a hypotonic solution, which means that there's more solute outside than there is inside. Okay? So let me try that one more time. My apologies. I misspeak sometimes. Okay, hypo, my apologies. Hypo means below. So the hypotonic means there's less solute outside than it is inside. There we go, I got it now. Because there's less outside than there is inside, okay, then where's the water at? Well, if we have less solute outside, hypo, if we have less outside, then we have more water outside. And if we have more water outside, osmosis, which is the diffusion of water, is going to make that water go from the outside in, just like this little arrow. And in an animal cell is going to cause that cell to swell and then lyse, okay? With the plant cell, the cell wall is going to maintain the pressure so the plant is okay. The plant can deal with a hypotonic surrounding. A plant can deal with an isotonic surrounding. It just can't deal with a hypertonic surrounding, okay? All right. So cells will gain or lose water relative to their surroundings in accordance with what the cell concentration is inside the cell as opposed to the outside, okay? A cell will lose water to a surrounding solution that is hypertonic because the solution has more solute than the cell. A solution that has a greater concentration of solutes in it than does the cell cytoplasm, okay? So it's important that you learn hypertonic, okay, more solute, isotonic, same solute, and hypotonic, less solute. Now, while we're in the topic, let's say you go to the hospital and somebody's going to give you intravenous fluids. What do you think you're going to take? Hyper, iso, or hypo? If you said isotonic, that is correct. IV fluids are isotonic to your cells because we really don't want your cells to shrivel or to lyse, right? Let's see. Water flow is balanced between the cell and its surroundings when the surrounding fluid and the cytoplasmic fluid are isotonic to each other. When they have the same concentration of solutes, replacement fluids, intravenous IVs, must be isotonic to other fluids. And I think that's where I'm going to stop. This thing won't let me stop. That's where I'm going to stop. And I'll be back in just a little bit. Okay? Thank you.